Hello, I'd like to welcome you to the Virtual World Stem Cell Summit meeting for 2022. I'm Dr. Jane Andrews, the founder and CEO of CellBridge Strategies, an international growth consulting firm that focuses on the life sciences and regenerative medicine. My team helps companies match their strengths with market opportunities by bridging the gap between science and business. Today, we're helping Bernie Siegel, the founder and director of the Regenerative Medicine Foundation and the World Stem Cell Summit. Today, the title of this talk is The Expanse, Session 2, Stem Cell Research on the International Space Station, Impact on Health, Aging, and Human Survival. So without further ado, I'd like to introduce the panelists. First is Michael Roberts. Thank you. That's Dr. Roberts. He is the Vice President at the Center for the Advancement of Science in Space, CASIS, and the Acting Chief Scientist of the International Space Station National Laboratory. As manager of the ISS National Laboratory in partnership with NASA, CASIS is responsible for enabling access for research, technology development, STEM education, and commercial innovation in space as a public service to foster a scalable and sustainable low Earth orbit economy, abbreviated LEO, low Earth orbit. Dr. Bill Wagner. Hello, Dr. Wagner is the director of the McCowan Institute for Regenerative Medicine. He is also a distinguished professor of surgery, chemical engineering, and bioengineering at the University of Pittsburgh. He serves as the scientific director of the NSF Engineering Research Center on revolutionizing metallic biomaterials. And he is the chief science officer for the Armed Forces Institute of Regenerative Medicine. Dr. Wagner's research interests are in cardiovascular engineering. It's clear that he is both a scientist and a businessman, and was induced into the National Academy of Inventors in 2017 and 2018. He is also actively involved in the space program and is a large contributor to the advancement of science in space, or CASIS. Arun Sharma. Arun. Dr. Sharma is a principal investigator at the Regenerative Medicine Institute, Cedars-Sinai Medical Center. His research focuses on the application of iPS cells for studying cardiovascular biology, modeling diseases in a dish with gene editing tools such as CRISPR and Cas9, and developing platforms for screening drug cardiotoxicity and efficacy. With regards to his interests in space biology, he has led a project that sent human iPS cell-derived heart cells to the International Space Station to study the effects of microgravity on human heart function. He remains actively involved in the space biosciences field and has received multiple awards for his work in cardiovascular iPS cells. So I think one of the first questions all of us would like to know the answer to, and I'll direct this to Michael. Michael, can you just go over the history of the National Lab and the thoughts behind its creation?
Yes, Jane, I'd love to. First of all, thank you for this opportunity and the World Stem Cell Summit as well. We look forward at the Center for the Advancement of Science and Space of being able to share opportunities to join us in space to further research and technology development that can advance medical outcomes here on Earth. And stem cells and regenerative medicine are certainly at the center of that. So speaking to the history of the International Space Station National Laboratory, as I'm sure most of you are aware, the International Space Station has been operating in space as a crew-tended platform since 2020. So we are now reaching 22 years of continuous human presence in space, humans working and living in the space environment. And throughout that entire tenure, even as the International Space Station was still under construction and being assembled in orbit, research, science experiments were being conducted in that environment. And A large focus of those early experiments were in understanding technologies to further um, humans' ability to live and work in the harsh environment of space, but there were also fundamental investigations focused on better understanding human physiology. And some of those of a more applied nature, seeking ways to identify mitigation strategies. So we understand that living in the microgravity environment has profound effects upon the human body. And some of those can be addressed directly through therapeutic actions. So you can combat the accelerated rate of bone mineral density loss and muscle atrophy by taking drugs. You can also combat those by exercise. Later on, through our history in space, we've also learned that in trying to understand the physiological responses in humans in that environment, those lended themselves to understanding conditions here on Earth, part of our normal aging process, part of disease onset and progression. So the International Space Station National Laboratory was created in 2005 by an act of Congress to enable NASA to engage with other federal agencies, other funding agencies in the United States, and to work with industry directly to support research and technology development in space that could leverage access to microgravity and low Earth orbit, but may not directly address the primary research goals that are funded by NASA and its goal to further explore space. So the ISS National Laboratory was created as an experiment to better enable access to space for agencies in addition to NASA and companies here on Earth. So from 2005 to 2010, the International Space Station National Laboratory was operated by NASA. In 2010, Congress wanted to remove that responsibility from NASA and explore the use of a non-governmental organization who could act with industry in a way that NASA was precluded from doing as a government organization. So in 2011, CASIS, as we say, the Center for Advancement of Science and Space, was offered the opportunity to assume that responsibility. So we entered into we were selected by NASA and entered into a cooperative agreement with NASA in 2011 to work with NASA to manage the International Space Station National Lab. So station's been in operation since 2000. CASIS has been in existence since 2011. And since 2005, the International Space Station National Laboratory has been an accessible laboratory operating in space to fulfill the needs of research and technology development goals here on Earth. So the purpose of that ISS National Lab is simply to provide access to tools and to that unique environment in space to further exploration goals that are important here on Earth. And as I mentioned in my introduction, regenerative medicine, health, aging, human survival are all key components of that research portfolio.
Could you give me a couple of examples of the types of regenerative medicine projects that have gone on station?
Certainly. So we work through our partnership not only with NASA, but with other government agencies as well. So throughout our years, we've enjoyed over 7 years of partnership with the National Science Foundation. As most of you are likely aware, the National Science Foundation research and development portfolio focuses primarily on understanding fundamental scientific principles. But it's valuable in the regenerative medicine space because we are able to leverage the National Science Foundation's expertise in engineering and biology and the intersection of those two. So through that partnership with the National Science Foundation, we've funded multiple years of research focused on the interaction of biological systems, microphysiological systems, bioreactors, in essence, that can be used for the culture of stem cells in that environment. We've also had an active partnership with the National Institutes of Health. The National Institutes of Health comprise several research institutes and centers, which are primarily focused on specific disease states. And their portfolio is, of course, focused on helping to mitigate the effects of disease by improving our ability to diagnose and treat and avoid disease in humans. But we partnered with two of those institutes, the National Center for Advancing Translational Sciences, or NCATS, and the National Institute for Biomedical Imaging and Bioengineering, NIBIB. Because unlike the other institutes and centers, they're not focused on a specific disease, they're focused on improving the tools that are available to address lots of diseases that affect us here on Earth. So through our partnership with NCATS and NIBIB, we initiated a program called Tissue Chips in Space, which continues to this day. And that enables access to space for NIH-funded research that utilizes microphysiological systems that are focused on disease modeling. These are small chip-sized devices that can be populated with induced pluripotent stem cells or primary stem cells. And if you are able to control their exposure to the environment and feed them in the right way, those small engineered systems can support populations of cells that mimic the physiology of entire human organs. So these are extremely useful as the technology matures for understanding the growth and maturation of organ systems. And they're very useful for pharmaceutical companies in the future because they enable us to expose human cells to test the efficacy and safety of therapeutic agents that will be processed through different organ systems. So these types of devices offer opportunities in the future for us to improve the outcomes of drug discovery and drug delivery mechanisms going forward. So we have a lot of experiments that have gone to space to take advantage of that access to the microgravity environment, which can act as an accelerant of disease onset and progression. We've had a lot of work with funding provided by National Science Foundation, National Institutes of Health, and NASA, of helping to improve the fidelity of the platforms that are used to cultivate these cells in that orbit. And then we've had a lot of support from the commercial sector. Large pharmaceutical companies as well as small companies have taken advantage of the microgravity environment to either test therapeutics of interest to their portfolios or to simply help them better understand the ways that they can grow and expand stem cell populations either for R&D use or for clinical use in the future.
It's very interesting to be able to use this platform where you experience accelerated aging and advancement of disease states to really test potential cures or methods for treating those diseases in a way that you really can't here on Earth. So it sounds like it's just an invaluable platform in those regards. Last year, CASAS, in collaboration with the University of Pittsburgh, McGowan Institute for Regenerative Medicine, put on a symposium to gather information about the types of life science experiments and regenerative medicine experiments that have been done on station, and look at them and look at the potential value proposition for some of those experiments. Can you tell me a bit about the symposium and how it came about and what that partnership was like and some of the key points or the outcomes of the study?
Certainly, yeah. So our collaborations and partnerships with stakeholders are extremely important. So we as a non-governmental organization working with NASA certainly understand our limitations. We don't have subject matter expertise in all the areas that can benefit from access to the platform in space. So we actively engage with the communities to identify not only subject matter experts in particular fields that have either utilized through partnership with NASA access to space to further R&D goals, but more importantly and more recently, we focused on organizations that have demonstrated the ability to translate fundamental research and technology development into clinical application and products that have real world value. So that led to our initial discussions with McGowan Institute for Regenerative Medicine, University of Pittsburgh, because of their demonstrated expertise in taking medical devices to market and moving beyond that to actually see them put forward for clinical application. So part of our mission at ISS National Lab is identifying technology advancements that can take advantage of the microgravity environment, but far more importantly, be translated from discovery in that environment to application here on Earth to improve medical outcomes. So partnership with McGowan was extremely important for that. And what we first set down to do is identify the shared value proposition between what University of Pittsburgh and McGowan Institute of Regenerative Medicine were about, and how that overlapped with our goals at ISS National Lab to advance the use of the platform to address these goals. So that enabled us kind of that next step, going beyond simply engaging with world-class investigators who were designing experiments for use on ISS National Lab. We actually had an opportunity to get in a room with experts who were not only looking at the outcomes of those experiments, but thinking about ways to accelerate their translation to clinical outcomes that would improve medical outcomes here on Earth. So it's a very important opportunity for us. We also brought in to that conversation elements that I think were new to that community of regenerative medicine, and those are the companies that have expertise in working in microgravity. In some cases, they have some insight into ways to miniaturize and automate systems that can increase to the level of throughput and lead to even further advancements here on Earth simply based on their prior exposure to designing hardware that will be operable in the space environment. So it's been a great conversation to be engaged in. We've been extremely pleased with the outcome to date, which has been a couple of white papers, one of them lead authored by Arun Sharma, who you'll hear from here in a minute. But we've been very proud of not only having that discussion, but in continuing the dialogue forward. So we continue to develop that relationship, and all of the stakeholders that are involved in that conversation are all focused in leveraging access to space to accelerate discovery here on Earth that can have real world outcomes.
Thank you. Thank you very much. So I'll direct this question to both Bill and Arun in regards to those white papers and the outcome and what the key findings were there with regards to the type of research, what's needed to bring these technologies closer to commercialization, really looking at how the science can become a business proposition. Bill, would you like to go first?
All right, thank you. Big challenge to try and summarize a lot of that work, but we looked for commonalities and broad potential impact, and ended up with a number of types of research and development that kept coming up. So certainly the first one was stem cell culture, proliferation under microgravity and the impact of microgravity on stem cell behavior and what stem cells secrete, and what value that secretome might have. The second was mentioned before, the microphysiological systems. So using the organ on a chip or coupled microphysiological systems that may replicate pathologies associated with various disease states in a relatively rapid fashion, and thus be able to be studied for druggability or reveal potentially new pathways. Maybe only related to microgravity, maybe directly related to terrestrial disease. The third area would be the biomaterials, self-assembly type processes. Most I'd say maybe furthest along right now would be adsorption processes of bioactive species, proteins, et cetera, onto, for instance, onto a sensor chip, and the ability to get higher fidelity in doing that under microgravity. So, self-assembly type processes that are susceptible to buoyancy type effects or other gravity-driven phenomenon that can cause problems on Earth. And then finally, I would say the 3D printing, bioprinting, and the big theoretical value is being able to work with a system where when it's laid down, it's not subject to the gravity force, and thus weaker less elastic, more viscous type behavior could be tolerated. Cross-linking procedures could be delayed, minimized, or maybe not even used at all. That would frankly open up the design space for what bioprinting might be able to do.
Interesting, interesting. That's fantastic. Arun, do you have anything to add there?
Yeah, I think Bill and Mike have both covered the opportunities quite well. Just to summarize, we focused really on these opportunities in disease modeling, stem cell derived products, and biofabrication, as Bill alluded to. I also wanted to emphasize that we're really in the nascent stages of a lot of this work. We still need to figure out what a lot of the beneficial fundamental mechanisms are of growing these cells and products in microgravity for translational applications here on the Earth. There have been a number of excellent experiments that have been conducted on the ISS and the US National Lab, focusing on stem cells and stem cell properties, and in some cases manufacturing as well. But a lot of these experiments still need to be repeated. They need to be iterated on. They need to be made more complex, comprising the multiple cell types that are found in the different tissues in the human body. So I think it's a really great start, but I still think we have a decent amount of legwork to do.
Which leads to the very next question. What are some of the gaps that you see, and what would facilitate getting over some of those gaps? I know there's a real need for additional data processing in low Earth orbit. There's additional IT capabilities, possibly blockchain, mechanization, automation, robotics. All of those things would really help advance the science, in which you were then able to get more replicates per experiment, so that you can really do some proof of concept work. So what do you think is the most important facilitative technology? I mean, or the three most important facilitative technologies that would allow you to move forward? Bill, do you want to take a crack at that first?
Yeah, I think, as you said, higher throughput, which means robotic processes for doing cell culture, not just the things you need to do to culture and maintain cells, but to monitor. So sensing, temporal sensing, specific marker sensing, aliquot collection and sensing.
Yes.
So building up a statistically robust data set that can be manipulated by artificial intelligence and transported back down terrestrially, so we can really truly understand. Right now, when you see the astronaut pipetting, that's not good. That means that you're limited to that individual, which is highly committed to lots of different tasks, doing that, doing it reproducibly and well. So having a robust robotic system to be able to collect that data, I think, is absolutely essential for many of the technologies that we're talking about.
That sounds exceptionally important. And then other things like data security, are you concerned about that?
To be frank, in our symposium, we didn't go into data security. We did talk about bandwidth issues and getting that amount of data pipe back down, and how we might be able to use AI models to minimize the bandwidth requirements, which are clearly at a premium. In terms of security, I think at any point when that data is being transported, you of course are worried about that. And security implies value, and that's, I think, the key recognition is that data now has profound value for potential intellectual property and future decisions. It's the first time that this kind of data will be able to be collected. So there should be security concerns, and there will be a lot of interesting intellectual property law firsts that will happen, I think, over the next decade, which should be interesting to see.
Very, very interesting. So robotics kind of goes along with automation. And one of the things we're facing here in biomanufacturing on Earth for regenerative medicine is the automation that's needed. So there's large pieces of equipment here on Earth that can treat the cells gently, or can help with expansion, or help with different aspects that give the cells the right mixture of gases, the right environment, so that you come out with the best cells possible without human intervention. And that we find here in biomanufacturing to be very important and still being developed. And I can only imagine the challenges, Arun, you must have in space, thinking about that automation, how to make that happen without human intervention, where you don't have an astronaut pipetting.
Yeah, absolutely. But I think really that's part of the fun, is to be able to think of some of these challenges and the unique ways to address some of these challenges. You're absolutely right. And I think a lot of the technologies that are Earthside have been advanced and developed, and in some cases perfected when it comes to automated cell culture, automated cell production. You know, in some situations, you can actually complete the entire induced pluripotent stem cell reprogramming process in a completely automated fashion, which is a 1 month long procedure with multiple steps. And what I'm thinking is, you know, we should be able to adapt some of these automated technologies for use in LEO as well. I think these are the same technologies that'll be helping us to answer some of those fundamental questions that I was alluding to that we still need to figure out.
Yeah.
To me, it makes perfect sense to adapt these ground side technologies to space.
Absolutely. Possibly miniaturizing them, I would think. Yeah. So, so the areas of research to be emphasized would be sort of that the organoid, or the organ, tissue, and stem cell. Are those the three types of cellular components that would be focused on? Sort of the organoids as well.
Yeah, I can take that. I think you know, there are three of a few different technologies that would be really interesting. I'm a basic stem cell biologist, so to me personally, organoid biology is definitely very exciting to pursue.
Sure.
I mean, just the concept of organoids in themselves are inherently three-dimensional structures that can more accurately recapitulate the three-dimensional architecture of different tissues that are found in the body. And in a microgravity context, there's some data out there that shows perhaps these cells can be grown better and perhaps proliferate in a slightly different way, in a more improved way, if they're grown in a suspension-based format, which is sort of similar to what you would see in microgravity anyways, right? When it comes to large-scale cell production, and in particular organoid production, bioreactors are really, you know, the way forward, in my opinion, for terrestrial production of these organoids and other spherical technologies, three-dimensional technologies. And I think that, you know, space and the microgravity environment could be another way to facilitate the production of some of these really exciting technologies.
That's wonderful. Another question I wanted to ask you, Arun, while I have you here, is how do we get more people such as yourselves involved in the Space Sciences Program, or CASAS?
Yeah, I think it's a great question. It's a very important one. I think I was very fortunate to be involved in some of this work as a graduate student, actually. It's an opportunity that not everybody gets to have. And I think part of it has to do with exposure, to know that these sort of opportunities are out there. And I think definitely the ISS National Lab has become much more accessible over the last few years. And I actually see a number of students and other prominent stem cell faculty and members of the community from around the world who are starting to recognize the potential and the power of the National Lab to further, you know, really unique research that just can't be done on the Earth. I think a lot of that has to do with leadership in our field, in the stem cell field, in the space biosciences field, in recognizing the importance of the next generation of trainees, because they're going to be the ones who are going to be completing some of these experiments and making some of these foundational discoveries that are going to bring some of these products to market that potentially are being manufactured in LEO. So I think, you know, to bring that next generation involved as quickly as possible is a very important thing.
Which kind of leads into the idea of the consortium, I would think, Bill. I know that you're putting together a consortium, and could you tell us a little bit about that and how that will help people like Arun and the next generation of scientists, and improving the outcomes of experiments on station, and the partnerships that'll be necessary, and, you know, who should take the lead, and how one step might lead to the next for advancing space science through a consortium approach?
Well, I think ultimately, like so much else in life, it comes down to economics.
Of course.
So when there's a expansion due to economic drivers of the low Earth orbit platform, in other words, there's frankly more square footage up there because there's a reason to be there. There's value that's being added in microgravity that can't be obtained on Earth. That then creates jobs. It creates, it grows a community. And as that community grows, just like a city, people go there. So I think fundamentally, there needs to be economic drivers, and government funding is necessary to overcome early stage gaps like we've identified.
Yes.
But also it needs to transition over to venture capital and private industry seeing it as valuable. So what we're trying to do in terms of a consortium is to bring to the table existing companies that already have identified an interest and potential value for their product or their area of emphasis in microgravity, and basically provide a forum and a mechanism by which those problems that are common can be identified and funding can be applied in a specific manner to help to overcome those challenges. So it's very much a public-private, government, industry, academia, where academia is able to do a lot of the research that's not as translational. Industry is identifying the key problems, and government is helping to apply resources at a magnitude early on in the process that can open things up later. And I think, again, as that all opens up and grows, that's where people see opportunity. And frankly, the one thing with space I've always found is it doesn't take a lot to convince people that it's cool and there's exciting possibilities, even if they themselves don't want to go up there. A lot of people actually do, but just to see that, hey, this is equivalent of the 19th century, gold rush. There's new possibilities. There's resources that we can access in a way that we weren't able to previously due to technology. So very exciting.
Sure. Anand, did you have something to add to that?
Yeah, I would agree. I think it's, like Bill was just alluding to, it's kind of an amazing time to be involved in this area, just because there's so much untapped potential, not just for building out that ecosystem, building out that regenerative medicine ecosystem, but just from just looking from the scientific perspective, there's just so many questions that are just awaiting to be answered. My dream is to find a cell state or a cell product or something that just cannot be done on the ground, right? A unique and inherently unique property that these cells can adapt just by growing them in space, that will have a tremendous return on the investment just immediately, right? If you have a completely unique entity and that's developed in space, the one question would be, how can you maintain that if those cells are returned back to the planet? That's, I think, a question that we'll have to answer in the future. But that possibility of just doing something that's not available on Earth is something that's very exciting to me.
Well, then biomanufacturing and obtaining the cell product. I mean, if all of the manufacturing could be done on the ISS through CASIS or through the consortium or a combination of those factors, with the advanced robotics, the IT component, the data processing, to do the biomanufacturing so that unique cell product can be created there in space, and then the product transported back and have useful applications on Earth. Is that sort of what you're thinking?
Yes, absolutely. I think that's really the ultimate goal, at least one of my goals. Scientifically, certainly, as I mentioned at the very beginning of the talk, there's a lot of legwork that has to happen before we can figure out that situation. But one other thing I think Bill is alluding to is that once we find that case situation where the science is good and it's beneficial, then there will be a domino situation in my view, and there's going to be an ecosystem that will explode around that discovery. So I think that's what we're really waiting for here.
Sure, sure. So I think there's we've asked a number of questions, and we've talked about the use of the ISS space station for advancing science in a number of different areas, and how biomanufacturing can occur. There's a need for additional technology so that we can do more repeatable experiments and then go closer to the commercialization process. It's all very, very important. And one thing often has to take place before another. So I think, as Bill pointed out, maybe some of this private and public partnering would help lead the way to the advancement of these efforts. Bill, what do you think needs to be done first?
Well, I think with this consortium, one thing that the government is interested is seeing buy-in from industry. It helps to legitimize the importance of the problem and demonstrate that there is, in fact, private equity that will be brought to bear if these important problems are overcome. So and industry allows us to really hone in on what they see as the critical needs from their business model perspective. So bringing them together, and then as a group working with government to then apply the resources, the accelerant that will address those problems, and then have follow-up. Now that that's overcome, indeed, is there going to be more private investment? Yeah, I think some of the markers ultimately are not just going to be private investment, but it's also going to be patent filing. Fundamentally, if you're discovering, and if there's proof of concept up here, it's going to need to be protected to attract maximum investment, and that will be a early marker of the growth in this future industry.
Thank you. That's a great summary. I really appreciate that. So I think I'm going to ask one final question of all three of you, and if each one of you could answer individually, what kind of impact do you think this work on the International Space Station will have on health, aging, and human survival within the next 5 to 10 years? Michael, would you like to take the lead on that?
Sure, I'll jump on that one. So in the immediate future, which is what I would consider the next 5 to 10 years, we are in a period of transition with the International Space Station. So we are approaching the end of life for International Space Station in 2030. And NASA has set forth its goal to establish what they are calling commercial LEO destinations. So speaking to a couple of the themes we touched on today, those are going to be commercially operated platforms in low Earth orbit that can sustain NASA's requirements for research in low Earth orbit, but can also support and will also support the research and technology development efforts, as well as more commercial activities of industry across the world, not just the United States. So we are at a very important crossroads where we are going to see a large infusion of capital from both the public and the private sector going forward. On the public side, NASA has put over $500 million into the design of these commercial LEO destinations to take over before station ends its operations. So that's a significant contribution to this platform. But more importantly, the companies that are developing these research platforms are focused on identifying new customers. So they're actively engaging with consortia to understand where the most advantageous research areas are that they can develop platforms for. Mm-hmm. They're starting to ask questions of funding agencies to understand where their research portfolio is going in the future. So while Arun and Bill are working very hard to accelerate the pace of discovery and clinical translation for these, there are others who are working just as hard to increase the fidelity of the platforms that are going to be necessary for them, because as Bill and Arun both correctly pointed out, we've scratched the surface. We understand enough to know that there is something very interesting about stem cell proliferation and expansion in the microgravity environment, but we really don't know what we don't know yet.
Right.
And we're going to need better resources. We're going to need a lot more data, a lot more capability to generate data rapidly in that environment to kind of not only address those questions that are so important for research advancement, but to understand how to better design systems to operate in that environment in the future.
What an exciting time, Michael. I think you're sitting in what a great place at a great time, where you're going to really be able to make a difference. As there's now some funding and some opportunity to advance those platforms to address the needs of doing more advanced research that will have human impact. Thank you. I appreciate that. Bill, would you like me to repeat the question?
No, I think I remember. 5 to 10 years impact.
Right.
And I think, realistically, I don't think that there's certainly an FDA-approved product that's being manufactured in space in 10 years. I do think, though, that, like I said, intellectual property, I think there will be exponential growth in that. And I think that there will be much clearer directions where public investment and private investment are beginning to move to really get more definition on organs on a chip and their value, or the specific aspects of stem cell behavior that might be leveraged. So really, primarily, I think what we'll see in 5 to 10 years is much more realistic business models that have detail within them that might be getting pitched and invested in to occupy space in the growing LEO platform that'll be out there in 5 to 10 years, and also setting it up for the next 5 to 10 years where maybe there will be a product that's manufactured in space in that timeline.
Well, thank you. That's really interesting. There's always, though, a fast track and orphan disease, so you just never know. I have to be optimistic, but anyway, Arun, I'm sorry. What are your thoughts?
Yeah, absolutely. I echoing what Bill and Mike are saying, I'm really optimistic about the next 5 to 10 years, in part because as a scientist, I think there are a lot of questions I think we're going to be answering when it comes to the fundamental biology.
Absolutely.
And my hope is that this ecosystem that we're talking about, this investment that we're talking about, is going to be founded on sound science. And I think that is what excites me the most. I think there's going to be case situations in cell production and tissue engineering where we're going to show definitively that that the microgravity environment, the spaceflight environment, can actually enhance the production of these tissues in a way that might be beneficial to various disease states and opportunities on Earth as well. So the science is what excites me the first and foremost. The other thing that really excites me, and something that we've alluded to a lot on this particular session, is the technology that's going to emerge from doing this science, whether it's automated processes, high throughput science, high throughput approaches. These are all technologies that we've perfected in a lot of ways on the Earth, and I think are going to be adapted to the microgravity environment and are going to facilitate really good science, which is the foundation of everything. And the final thing that I'm really excited about is, again, something we alluded to. It's the educational opportunities. We're going to have an entire group of students who will be able to see that research in space is real. It's something that you can found your career on in some situations and devote yourself to. And I think that is also a very important point and takeaway, that, these are the students, the educators, the folks who are going to be doing this, not for the next 5 to 10 years, but for many years to come. And so that's what really excites me.
Yeah, that's very exciting. It really is. And as I've learned over the years, if you don't have the science, you have nothing. It's the science that everything else is built off of. So I really like your perspective as well. Well, thank you very much, Michael, Bill, and Arun, for participating in this World Stem Cell Summit webinar. It's been invaluable and I think very educational for all of those that view it. And thank you again very much for your time.
Thank you.
Thank you, Jen.
Thank you.